Let's actually now look at the technique itself of gel electrophoresis or DNA electrophoresis and how it works. What is the principle behind it? So how can we basically use it to separate DNA fragments according to size? It uses the fundamental property of DNA, which is that because of the phosphate groups that it contains on its backbone, DNA as a molecule has a slight negative charge, meaning that if I put it into a solution that contains electrolytes, and I apply an electric field here, my negatively charged DNA is going to be attracted towards the positive pole. So what does electrophoresis require in order to be set up before we actually see the step-by-step process? It requires a digested DNA sample. Why do we say digested? Because we cannot take an entire chromosome that is millions of base pairs long. It's just not a size of DNA that we can work with this technique. So when we say digested, we mean that we did what we said previously. We use a restriction enzyme, a restriction endonuclease, with the appropriate recognition sites before and after the region that I want to cut out the gene of interest that I want to, or the VNTR, the microsatellite of interest that I want to study. So I need an actual agarose gel. Agarose is a type of, literally a gel, very similar to the gel that you eat. It's extracted from a type of seaweed. Comes in a powder, you put it in liquid, you melt it in the microwave, and then you pour it into this kind of like plastic container here, and it solidifies when it cools down, exactly like gel. We also add a little kind of like a plastic comb inside the gel as it's setting when it's still liquid, and we then take it out. And that creates little pockets. So if you were to look at the gel from another view, you would see here's my gel. That creates little pockets for the DNA to be able to sit in. I then also use something called a loading dye, which is a pigment, just a simple pigment that I mix with my DNA sample so I can actually see it physically. I can see it going into these bands. This is why you've got these dark blue little wells here. A DNA ladder, we're going to explain exactly what that is in a minute, and an actual electrophoresis tank. So a setup like that, basically with an electrical current across it. You connect that to a power source, of course, and the current runs. Let's see what the process is on a step-by-step method. So we first of all add the loading dye, which is said as a simple pigment. So you could, because DNA is transparent, the DNA solution is transparent. You can't really see it when it goes into the gel. We add it to our PCR tubes or to our restriction endonuclease mixture. And we pipette it, meaning that we use a micro pipette that can dispense very small volumes of the order of like, anything between like 10 and 1,000 microliters. We put it in the wells of the agarose plate along with a DNA ladder. Now, what does the DNA ladder do? The DNA ladder contains a mixture of DNA fragments of known size. What does that mean? It means that, so for example, it's going to contain our DNA fragment that is, let's say, 1,000 base pairs long. It's going to contain another fragment that is 500 base pairs long, and another one that is going to be 100 base pairs long. The 100 fragment is going to migrate further than the 500 than the 1,000. So I know that, and here I'm going to have my mixture of my unique banding pattern that I'm going to get from my actual DNA samples. So say that a sample runs here, another sample runs here, another sample runs here. I will be able to look at the distance between my sample and the ladder and say, oh, well, if this 1000 base pair piece migrated this far, and my piece has run a bit, so it looks like my piece might be around 800 base pairs, for example. This piece looks like it might be around like 400 and so on. An electric current is then applied across the gel and it causes the DNA to move to the positive anode. The positive end is going to be down here. I load my DNA up here and it's going to migrate this way because negative is attracted to positive, of course. The smaller fragments, of course, move faster and they travel further in the same period of time, whereas the larger fragments will travel slower. This is because the agarose gel, if you were able to see it with a microscope, it looks almost like a sponge. It has like a porous, so the DNA has to kind of like move in between the sort of spongy structure of the gel. And so if you're smaller, you can move faster. So the reason is because of less resistance. I also add a separate, so don't confuse the DNA binding dye with the DNA loading dye. The DNA loading dye is just the pigment that shows me whether my DNA went into the well. Did it actually go into the well? The DNA binding dye is usually a fluorescent type of dye that actually physically binds to my DNA, and it will enable me to visualize these bands here. It'll enable me to visualize how far my DNA has run in the gel. Let's see an example question. So DNA profiling can be used to compare the genomes of different people. It is used in forensic for paternity tests. Explain the role of gel electrophoresis in DNA profiling. So my context is that I am looking at DNA profiling and gel electrophoresis. This is the part of my spec. My command is an explain, so a more detailed response, explain. And my direction is that I have to define what DNA profiling is, and I'm going to have to also explain the electrophoresis and link the two together, link the two concepts together. Take a couple of seconds to jot down a quick answer plan. So the role of gel electrophoresis in DNA profiling, well, it's used to separate fragments of DNA by size, thereby generating a readable fingerprint or banding pattern, you can say, that is unique to each individual, second mark. Different DNA samples can be compared to find commonalities in the gel bands. So we are also stating, how do we use the results that we get from DNA profiling?